All right, why don't we start? I guess a huge crowd is not coming all of a sudden. So in this lecture, what I cover are molecules that are involved in antigen recognition, and most of those molecules are involved in the acquired immune response. Okay, we'll talk about the acquired immune response when we talk about T-cell activation and antigen and processing and presentation. So those are molecules that, at least for immunoglobulins and T-cell receptors, their specificity against particular antigen is continuously changed and randomly made. Okay? Now that branch of the immune system is the one that kind of remembers what the antigen is, mounts a stronger response when that antigen comes from a second time. But you also have a branch of the immune system that does not change continuously, okay, but rather is fixed in its repertoire of specificity, and that's the innate immune system. So innate immunity is very important, especially in initial exposure to a particular antigen or pathogen. Okay, I mean, just to give you an idea, if you're failing in an innate immune system, you have a lot of problems. This is like an extreme case of oral candidiasis, okay, thrush. In this case, I think it's from an AIDS patient, okay. So your body has a system that learns to adapt to certain antigens, okay, but also has another system that has the approach of be prepared for any possibility, okay. Be prepared, like a good Boy Scout. Now, there are functional differences between an innate and adaptive immune response. You may sometimes, especially in older books, find that the innate immune response is also called nonspecific. That's really a misnomer, because obviously the response has to be to a specific something. It's just that the difference is that the specificity is much more promiscuous, it's much more broader, much more larger than an acquired immune system, okay, because it looks at patterns rather than specific molecules. So in general, an innate response is usually the first response to some sort of insult, okay, some sort of injury or attack. It's mediated by myeloid cells, has some specificity, but does not have any memory, meaning that each time an antigen encounters the innate immune system, the innate immune system treats that antigen as it's seen it for the first time, even though it's coming a second time, okay? Where the acquired immune system kind of remembers that antigen. Okay, on the other hand, the adaptive response, or the acquired response, is mediated by lymphocytes such as T and B cells, has memory. It's highly specific, I mean it can distinguish very minute differences between an antigen and another. Okay, but it takes a long time to get started. So in general, when you have an insult, you have an initial wave of response that is usually the innate immune system. Okay? So here are the difference between innate and adaptive immune system. The time, as you see, okay, is much faster in an innate immune response than an adaptive immune response. Although the adaptive immune response, once kicked in and remembers it, okay, can be faster, okay, and can be stronger. Now, the key difference here is the adaptive immune system improves during response, whereas in the innate immune system, it's limited and it's fixed. Again, you don't create novel specificities like you do for the acquired immune system. Okay? The strength of the response is usually identical to primary response, whereas an adaptive is much stronger than primary response. And these are the components that are associated with these two different branches of the immune system. Okay? Now, the genes that are involved in the components that participate in these immune branches are present at birth, but in the adaptive immune system are altered after birth. And we went through the mechanism by which these specificities are randomly created. So the first line of defense against a particular insult is usually trying to keep things out. So you may argue whether this is actually an immune phenomena or not. Okay, but in general, your initial protection against mechanism are the walls of a military force. Okay, skin, enzymes, physiological responses such as sneezing and coughing is to try to keep things out. Okay. Things like, the hair in your nose, not the hottest thing on first date, but you know what? It's the side to keep things out. Okay, so the initial responses are usually physical barrier, physiological responses, and things like mucus, okay, cilia, things like that. So what happens if these barriers are now breached? So now you have a situation in which you have some sort of tissue damage. This is, I guess it's a blade or something, okay, that cuts through and introduces an insult, such as bacteria. Okay? Let me just... All right, so the first initial response are usually an inflammatory response, which in turn causes the release of a whole bunch of different vasoactive and chemotactic factors, okay, which induce the first initial spurt of inflammation. Now, this in turn induces various different functions. For example, it increases capillary permeability. Okay, and why do capillaries become more permeable? Because they allow phagocytic cells, okay, the soldiers of the immune army, the Marines of the special forces of the immune army, to go through to go to the site of infection. And of course, there is release of chemotactic factor. Chemotaxis is attracting a particular phagocytic cell to the site, okay, where you have this, where neutrophil, or I call it the happy stone macrophage, okay, right here, come to the site and do their job. Okay, now these are the cells that are involved in an innate immune response. Now some of them are actually involved in both innate as well as acquired. Okay, but basically you have neutrophils. Neutrophils are basically, think of it as the Marines of the immune army. They're the first wave of soldiers to the site. And many times they actually act as kamikaze soldiers. They go to the site and sometimes they extravasate all kinds of bad chemicals, reactive oxygen species chemicals, things like that, that in turn damage the attacking bacterium. And in many times they also damage the tissue on which these microorganisms are laid. Macrophages wear many different hats in the immune system. They can be part of the innate immune system because they're phagocytic cells. They also can turn on the acquired immune system because, as we'll see in a different lecture, they also are antigen-presenting cells. Dendritic cells are the major antigen-presenting cells. These guys go around scooping stuff up all the time, looking for foreign materials, breaking it down and presenting these foreign materials to awaiting T cells, okay? We'll have a whole lecture on antigen presentation, but briefly, what happens when an antigen presenting cell presents to a T cells being the officers of the immune army, is that the soldier in the field captures the enemy and tears them apart, physically tears them apart. Then goes to an awaiting T cell, which is the officers of the immune army, and presents pieces of that enemy. This is his bleeding head. This is the weapon that he's using. This is part of his uniform. This is a piece of the flag that he was carrying. The T cells now responds to the antigen presenting cell and says, yes, indeed, I agree with you that this is the enemy. Therefore, I further instruct you to go forth on your attack. And we'll cover the whole procedure of this in a different lecture. And then you have natural killer cells, which are usually cells that are involved in the detection of viral infected cells or certain tumor determinants, and we'll cover those. Okay, so neutrophils are usually the first line of defense against a particular insult, okay? When you have a pimple on your nose, the white stuff in it contains a lot of neutrophils, okay? And what they do is they are recruited or to the infected or wounded site, where what they do is they try to destroy the initial attacking organism, okay, by undergoing phagocytosis and killing them, okay, by creating bags of digestive material within the cell after ingesting the microorganism that in turn destroys or damages that particular microorganism. Okay? Now, how you recruit neutrophils is by a process called chemotaxis. Okay? Chemotaxis is a process by which leukocytes detect a certain chemoattractant, detect certain smells, and move toward a concentration gradient of those smells. Now the smells, these chemicals, chemoattractants, are either derived from the offending organism, or they are derived from the tissue that is under attack and secretes these chemoattractants to call its soldier to the side, come and help me, okay? So the way it works is that, imagine that somebody here is cooking lasagna someplace, okay? I say lasagna because I got to cover my heritage, okay? So what you do is you smell the lasagna and you do what? You physically move toward an increasing concentration of lasagna smell, simple as that, okay? Until you get to the place where the food is being cooked. Well, neutrophils and other chemotaxic cells do exactly that. They have noses for these particular chemicals, and they are able to locomote, physically locomote, to the site where these chemicals are released. Okay? Now, what they do in order to do that, and this is a very important point, the way immune cells go from here to there is what they do is they express certain molecules on their surface, called adhesion molecules, that jive chemically, that bind chemically to sticky molecules on the surface on which they walk. For example, the endothelial barrier on a blood vessel. Okay? So imagine an astronaut in free gravity in space, no gravity, that can walk on a surface because he or she has Velcro shoes on a Velcro floor. So, you know, the Velcro's got the furry part of the floor, and the shoes has the hooky part of the Velcro, and he simply sticks and walks. Okay? This is how cells do. Now, the reason this is very important, because there are diseases, and we'll talk about that when we talk about immunodeficiency, where some of these sticky molecules are defective, and so what happens is phagocytic cells cannot stick to the floors on which they walk, and therefore cannot arrive to the site of infection. In other words, they find themselves having bald tires on glare ice. They keep spinning, but they cannot go to the site of infection. So the 4 steps in which neutrophils are activated are a rolling adhesion, an activation, an arrest, and then an extravasation. And I have a picture of it. So here is a neutrophil just happily jiving down the bloodstream. And all of a sudden, there's some source of insult that is detected, and inflammatory mediators are released. And now the neutrophil loosely adheres to the endothelium, okay? This is the inside of the vessel. This is the outside of the vessel, okay? And starts to stick. As the insult gets more and more detected and more inflammatory mediators or chemoattractants are released, then the neutrophil sticks firmly and eventually squeezes through the endothelial wall to arrive to the site of infection. And this is catalyzed by a whole bunch of different of these sticky molecules, okay, which are present on the neutrophil as well as on the endothelium, okay? All these molecules are crucial for the neutrophils to stick to the endothelium and to go through the extracellular matrix, okay? If you have problem with those molecules, once again, we'll talk about these pathologies, okay? Your neutrophils and other phagocytic cells cannot arrive to the site, and you have issues with infections. Also, if some of these neutrophils' mechanism to digest particular microorganism are defective, then you also have pathology. For example, you have in chronic granulomatous disease, you have a defect in respiratory bursts. Respiratory burst is a biochemical event that phagocytic cells, particularly neutrophils, develop to make all those bad chemicals that in turn kill and destroy the microorganism after it's been ingested. So what happens is this disease, because you have some metabolic defect, okay, it's a defect in the NADPH oxidase. I don't know if you guys have biochemistry or not, if this means anything to you, but is one of the enzymes that is used to catalyze the production of these very reactive chemicals that in turn are used by the neutrophils to kill the bugs, okay? And what happens? What happens, they can ingest the bugs, but they cannot get rid of them. And so more cells come to the side, more phagocytic cells come to the side, and you get the developments of granulomas. So the second set of innate cells are macrophages that also are involved in phagocytosis. They also release a lot of inflammatory mediators, various different cytokines that in turn exacerbate inflammation. They also are antigen-presenting cells. Once again, we'll cover that in the next lecture. Okay? They do also, like neutrophils, produce reactive oxygen and nitrogen species, which in turn are used to kill the bug and digest it. Okay, and they release various different cytokines, which in turn turn on other cells. Cytokines are chemical mediators that are used by a cell to communicate with another cell. Think of it as the radio signals of the immune army. Okay, a cell A secretes a certain cytokine, cell B receives that cytokine, and that action in turn dictates what cell B does. Okay? And here is a diagram, I'm sure you've seen this in basic biology book of phagocytosis, where you have an internalized bacterium. Okay, the bacterium is ingested into a phagosome, and now you have these bags of very bad chemicals, digestive enzyme, oxygen, reactive oxygen and nitrogen species, which then are emptied into the lysosomes, into a lysophagosome, and then the microorganism is now destroyed. And this, think of this as the burp stage, which, you know, it's done, and now, you know, you excrete stuff that is harmless. Okay? Now, somewhere in the line it doesn't show here, some of these cells save some of these pieces for antigen presentation, which we'll cover next week. Okay? And this is just a pretty picture from Kubi showing a phagocytic cells attaching to particular microorganisms. Now, when we talked about the acquired immune system, we talked about antibodies, we talked about T-cell receptors and how they're able to detect an antigen. Okay? Now, innate immune cells, in order to react to a particular insult, to an organism, for example, they also have to be able to recognize them. Okay? This is where the old name, nonspecific immunity, is really a misnomer, because the cell must know that this is something foreign. Okay? And once become activated, they do all kinds of different things. Okay? They increase phagocytic activity, they increase engulfing of microbes, they secrete inflammatory cytokines, et cetera, et cetera, et cetera. So the key issue here is, if the acquired immune system uses antibodies and T-cell receptors to recognize an antigen, how does the innate immune system, which does not have these kind of receptors, recognize something that they deem to be foreign? Okay? And this is the key. Patterns. Okay? If you see this dude here on the highway hijacking, I ain't going to stop. Why? Because he exhibits a pattern that suggests he's one bad guy. Okay? Okay? Now, for all you know, he could be a Baptist minister from Oklahoma, okay, coming back from a woodcutting contest. But chances are, because of the pattern that he shows, it poses a potential threat, okay? So the innate immune system sees more of pathogenic patterns rather than very highly specific molecules. Now, what detects these pathogenic patterns? Well, there's a whole bunch of different receptors, okay, that are involved in the innate immune system. And once again, these receptors have a little broader specificity. They have more promiscuous specificity. In other words, they can recognize more than one pattern. Okay, the acquired immune system has exquisite specificity. You change your carbon atom on a molecule, they can detect it. Okay, whereas this is more broad patterns of recognition. Okay, and these are a bunch of different innate immune receptors. The most common ones are called toll-like receptors. Now, toll-like receptors are receptors that are called such because when people were studying fruit flies, Drosophila, okay, there were some receptor in the fly that was shown to be necessary for fighting infections of the fly. And they were called toll receptors. So because these receptors in humans have similar function, historically they were called toll-like receptors. And what happens is they are receptors that are usually involved in microbial components that are not found in host. Okay? So if you have a component that is usually not known to be a mammalian component, well, then it must be bad. Once again, the immune system doesn't know what's good or bad for you, simply what belongs and doesn't belong. Okay? Now, there's a whole bunch of different receptors. Okay, and what they do, they recognize patterns of pathogenicity, things that are usually associated with a bacterium, for example. Okay, now toll-like receptors differ in their function because some can recognize extracellular pathogens, while others can recognize intracellular pathogens. So some microorganisms attack you on the outside of an immune cell. Some, on the other hand, are intracellular organisms. I'm sure you must have covered some in this course, okay, such as viruses or certain bacteria that have to physically enter the cell before they have their infective or their life cycle. Okay, so here are examples of mammalian toll-like receptors. Okay, TLR2, which recognizes a lot of different bacterial components. For instance, lipopolysaccharide, gram-positive features of gram-positive microorganism, certain lipoproteins, all these that are characteristic of a microorganism. Okay, and here's TLR4, which also recognizes various different bacterial components. And here's, for instance, a particular toll-like receptor that recognizes internal components, such as certain bacterial DNA. Okay? Okay, these are toll-like receptors that are capable of recognizing viruses. Okay, and these are usually inside the cell because the virus has to enter the cell and then turn on the mechanism by which they reproduce. Okay, so for example, this particular one, and no, I'm not going to ask you to remember does TLR2 bind, this particular thing. Just get the concept. Okay, for instance, this particular receptor recognizes double-stranded RNA of certain microorganism. Okay, others recognize other intracellular organisms. So some toll receptors recognize patterns that are derived from extracellular sources, while some recognize patterns that are derived via intracellular sources, for example, viral components. Okay. Now, there are some other receptors beyond toll-like receptors, again in the immune system, such as NODs, or nucleotide binding oligomerization domains. And what they do is they recognize cytosolic components that are derived from microbes that have to enter the cell in order to infect. Okay? And what happens is that you have a various different cascade, which in turn causes the release of various different components that fight that particular microorganism. Okay? And of course, these are very necessary because if you have mutation in some of the genes that encode for these components, okay, you have pathology. We'll cover these components when we talk about autoimmunity and hypersensitivity. Okay? In addition, some cells can recognize the presence of replicating viruses, even if they don't have toll-like receptors. Okay? For example, certain RNA helicases, which are involved in viral life cycle, feed into what is called the NF-kappa B pathway. NF-kappa B is a nuclear factor that turns on many genes that are involved in inflammatory and immune responses. So even if the virus gets in, and even if it bypasses the particular toll-like receptors that are designed to detect that virus, there are other components within the cell that can also activate inflammatory pathways that in turn result in some response. Okay? Now, depending on the situation, you can have either local versus systemic responses. In general, what happens is local inflammatory responses are mild and short-lived. Okay? So once again, you have a very small insult, a breach of the skin, you got an introduction of a microorganism, you get, a pimple, or you get, a red inflammation area, but the thing is resolved fairly quickly without a lot of different, pathology systemically. However, if the innate immune system cannot contain that particular insult locally, and it goes systemically, now you have a whole bunch of other mechanisms that kick in. Okay, and these are systemic responses, and in certain cases they have more extensive consequences. Okay, sometimes there are such great release of certain cytokines, for instance TNF, that you can actually get into septic shock syndrome, or things like that. Okay, so usually the tie between a localized and systemic involves the production of various different cytokines. So you have an inflammation and localized response. Okay, the inflammation process leads to production of inflammatory cytokines. Once again, these are chemicals that these inflammatory cells release to send signal to other cells to either come to the site or to activate their other mechanism. Okay? And what they do is, once again, either they recruit other cells to the site, come and help me, I need help, I need more soldier to the site, okay? And also, if it gets out of hand, they send signals to systemic systems, okay, if the local containment doesn't work. And this, in turn, generates what is called an acute phase response. An acute phase response now goes systemically. So instead of having the pimple on your finger, now you have some sort of, let's say, bacteriemia systemically. Now you kick in a much broader response, okay, because it affects many different parts of the body, not just the site of infection, which in turn is fever, very classical response to systemic infection. Okay, and also the production of various different hormones, okay, for instance, ACTH. And then in this response, the subsequent step is the liver producing acute phase proteins. And acute phase proteins are the next wave on an innate response when a system goes systemically. And these are examples of acute phase proteins, C-reactive proteins. Complement, we'll complement when we talk about effector mechanism. Fibrinogen, all these are involved in the amplification of a response that now is going systemically. Okay. In addition, you have the release of various different inflammatory cytokines, okay, that turn on the acute phase response. So, for example, if you have an insult at a particular site and it cannot be contained, now the cells that deal with this particular insult start secreting inflammatory cytokines that go systemically. For instance, interleukin-1, TNF-alpha, interleukin-6. And this, in turn, as a second wave, turn on the acute phase response. Okay, and here's a picture of the whole thing. So you have a local acute inflammatory response. The inflammation is not able to contain the insult. The insult or the attack gets stronger and stronger, and now you have an expansion of the response. For example, you have cytokines, inflammatory cytokines, that hit the hypothalamus, which in turn, we have the pituitary releases ACTH, which in turn stimulates the adrenal cortex to release corticosteroids, which in turn go to the liver that produces acute phase proteins. Okay, the same cytokines also directly go to the liver to stimulate acute phase proteins. The same cytokines also can go to the bone marrow. The bone marrow is the source of the immune cells, to tell the bone marrow, please make more of this kind of an immune cells, send more of these kind of soldiers, because I need help at this particular site. So the whole system then generates what is called an acute phase response, which in turn produces acute phase proteins such as these guys right here. Okay? This is all part of a systemic innate immune response. Now, one of the acute phase proteins, there's different ones, are C-reactive proteins. C-reactive protein is a protein that basically is used nowadays to reflect ongoing inflammation, and actually it is used as a diagnostic value to detect, potential inflammation. It's a protein that kind of prepares a little niche for complement to be activated. Okay, we'll talk about complement in a different lecture. And now it's clinically used to detect various different inflammatory conditions. For example, bacterial endocarditis, okay, neonatal septicemia, and post-operative complications. So even though it's an acute phase protein that does its own job, it's usually used diagnostically to analyze potentially inflammatory conditions. Now, in addition to all these wonderful players, you also have various different antiseptic or antibacterial proteins, small proteins, peptides, that are present in various different areas. For instance, in your saliva. Okay? These are highly conserved, evolutionally, these are highly conserved proteins that are present in many different types of animals, actually plants too, frogs, flies, humans, plants, a whole bunch of other stuff, okay? And these are used as antibacterial, okay? If you ever wonder why a dog licks his wounds, okay, part of it is because saliva has certain antibacterial properties. Okay, and these are examples of antimicrobial peptides. And no, I'm not going to ask you where do you find catalysid or these guys right here. Just know that you produce various different peptides, okay, that non-specifically, broadly, also have antimicrobial properties. In humans, for example, you find the alpha defenses and the beta defenses, okay, which is found in epithelia and other tissue, cells of the intestine, also granules of neutrophils. In addition to those highly reactive oxygen and nitrogen species, neutrophils can also produce these guys to further help them kill a particular ingested microorganism. Now, there's a whole bunch of different cells that are also involved in the innate immune system. One of them is NK cell, or natural killer cells. Now, natural killer cells are usually designed to target and kill virus-infected cells and sometimes certain tumor cells. Okay? So their specific job is to recognize various different viral determinants or various different tumor determinants, and to destroy those cells that express these determinants. So a viral infected cell will express viral components, okay? NK cells are able to recognize these viral components and kill the cell that is infected by the virus before that cell has the chance to butt out the virus that infects other cells, okay? Now, I'm going to skip this because this is dependent on understanding antigen processing and presentation. But just remember that NK cell also inhibit receptors that prevent killing in the presence of major histocompatibility complex. We'll cover that in the next lecture. Okay? So what they do is they basically guard against virally infected cells. They're not the only ones, okay? And also certain tumor cells. And finally, dendritic cells. Now, dendritic cells, their major job in dendritic cells is to constantly present antigens to awaiting T cells. Once again, we'll cover that in the next lecture. These are the guys that are also called professional antigen presenting cells because that's their major purpose. Okay, macrophages are also antigen presenting cells, but they also do a lot of other stuff. Where dendritic cells are the major link between the innate immune system and the acquired immune systems. Those are the guys that continually swoop things in, and anything that is deemed foreign is broken down and presented to awaiting T cells. And what they do is they link the adaptive and the innate immune system by presenting antigens to the cells of the adaptive or acquired immune system. Okay? Now, there's a whole bunch of other ones, but again, you have to remember that I'm limited by the amount of stuff that I can present in 7 lectures. But just to show you, there's a whole bunch of other stuff, other components that contribute to inflammation. Okay, fibroblasts, epithelial cells, all those have some sort of TLRs, and of course cytokine receptors. Okay, and the common pathway of what these guys do, they turn on the NF-kappa B pathway, which in turn is the pathway that turns on the genes that encode for inflammatory cytokines. Okay, so these are the genes that are turned on by the NF-kappa B pathway. Okay, again, I don't have time to cover this in detail. So basically what happens, there's an interplay between the innate immune system and the adaptive immune system, and we'll cover the major component of the interplay in the next lecture. Okay? So you have toll-like receptor that recognize component of microbial pathogen, mostly patterns, similar patterns. Okay? So there is promiscuity among receptors. More than one receptor can recognize a particular pattern, and a single receptor can recognize more than one pattern. And this in turn then triggers the release of various different inflammatory cytokines, for example interleukin-1, and also upregulation of various co-stimulatory molecules which activates T cell. We'll talk about that in the next lecture. Okay? In addition to this, what they do is they also ingest the bacteria, kill it, and then present pieces of this bacterium to an awaiting T cell. Okay? So basically, there's an interplay between the innate immune system and the acquired immune system. The innate immune system being a first wave of defense against something that has breached those barriers. The elimination, at least try to eliminate this particular offense, okay, while telling the acquired immune system, hey, listen, I just got this guy, I killed him, but this is what he looks like. So remember, because when the time comes, then he shows around again, we have to mount a stronger response. And we'll cover this mechanism in the other lectures. Okay, so if you don't have any question, my grandma was hot. Okay, if you have any question, let's go eat.